Today we will be discussing the interaction and individual effects of diet and light intensity on anxiety, risk-taking, and cognition in Nile grass rats. We will be questioning how obesity, hypertension, and type 2 diabetes are affecting behavior disorders. As you can see, the obesity trends in the U.S. are on the rise. As mentioned before, we are going to be seeing how anxiety, risk-taking, behavior, and cognition and spatial learning is affected with the diets and the lighting conditions that are given. We are doing this, we are testing the anxiety behaviors through the open field test, and by doing this we can see their anxiety levels and how much time they spend on the outside and the inside of the box. We are testing the risk-taking behavior through the elevated plus maze, and how much time is spent in the center zone, on the open arms, and on the closed arms. And lastly, we are testing the cognition and spatial learning through the Morris water maze, and how well they can find their way back to the platform that they started on. Okay. The purpose of this study is to further our knowledge on how diet and light intensity can affect anxiety levels, risk-taking behavior, and cognition function. We are doing this because obesity is a growing problem in the U.S., and this study can give insight to the magnitude of effect in diet and light that have on human behavior. While we are doing this in Nile grass rats, this can also be applied to how it affects people in later studies. Like I said, we are using Nile grass rats, which are diurnal species. The hypothesis that we are testing is that a high-fat diet and dim lighting will result in a compounding effect that will heighten anxiety, risk-taking behavior, and cognitive impairments.
As far as the methods discussion, we started with 27 total Nile grass rats. 13 were males, 14 were females. Each of those grass rats were individually housed and treated randomly. The first treatment group included 5 rats with high fat and dim lighting conditions. The second treatment group had 9 rats with high fat and dim lighting conditions. The third treatment group had controlled diet with dim light, and that included 5 Nile grass rats. The last group was the control group with controlled diet and bright light conditions, and that included 8 rats. The controlled diet contained 9% fat content that was complete rat chow. The high fat diet group contained 19.6% fat, and that contained all the goodies that we love: Ruffle chips, potato chips, Chips Ahoy cookies, peanut butter, and chocolate flavored Nesquik powder. The treatment group for the lighting condition, the dim lighting condition, had 50 lux, and the bright light condition had 1,000 lux. Over the course of 3 weeks, we completed 3 different maze courses with the Nile grass rats. In the first week, we completed an open field test, and the rats were exposed to the open field test, which was a box with no lines, and we watched them using an Any Maze behavioral test tracking device, and we analyzed the amount of time that they spent in the center zone versus the perimeter zone. Each rat was tested for 5 minutes total for one trial. The second week, we used the same tracking device, and we used an elevated plus maze. We analyzed the amount of time they spent in the open arms versus the closed arms, and even the center zone. The center zone is the area between the open arms and the closed arms. The final week, we used a Morris water maze, which had visual stimuli placed around the water pool, and it had white paint included in the water pool with a hidden platform. The rats were tested for 60 seconds and their goal was to find the hidden platform. This tested memory and spatial learning. The following results from each of the studies were pretty interesting. The first result that we concluded was that there was 26.6% greater body mass in the rats treated with high-fat diet than normal diet. This was vital to our experiment because it proves that the high-fat diet does statistically significantly affect the makeup of the rat. So any further results can be concluded back to this as the primary source. The results from the open field test had an interactional relationship between lighting treatment and diet treatment. The control diet and bright light had statistically greater time spent in the center zone than the high-fat diet with bright light. The controlled diet and bright light also had statistically higher time spent in the center zone than the controlled diet and dim light group. So from this we can understand that the controlled diet and bright light group were less anxious due to spending more time in the center zone than the perimeter. The perimeter area can be understood as anxious behavior from the Nile grass rats due to wanting to be near the edge, and that's understood as the comfort zone. Also in the elevated plus maze, it is understood that with dim light, rats spend more time in the closed arms. This is about 37% more time spent in the closed arms when treated with dim light. This is understood as an anxious behavior because the closed arms are, again, the safety area. They feel more safe. And so light treatment can also affect the amount of center entries. With bright light, there is a 49.2% greater entries to the center zone. This is understood that bright light can increase the risk-taking behavior due to the increase in travel between the arms and the increase in decision-making or lack thereof of where the rats want to travel. Light treatment in the center distance. This is pretty much the same thing. If they're going to have more entries to the center zone, then they're going to travel more in the center zone. But this can be understood as spatial awareness, the ability to make decisions based off what arm they want to go to, closed or open. Lastly, for the elevated plus maze, the lighting treatment can affect the amount of time spent in the open arms. So when treated with bright light, there's a 65.4% greater time spent in the open arms. This shows that with bright light, the rats are a lot more capable of displaying risk-taking behaviors and lack of anxiety behaviors due to the ability and the comfort to be in the open arms. Lastly was the Morris water maze. And unfortunately, we did not have any statistically significant results with this test, but we had results that trended towards a decrease in average time finding the hidden platform when treated with dim light. This may be understood because rats treated with bright light are more risk-taking, so they don't have the amount of motivation to find the platform as the dim light rats did.
Our reasoning for this study was to determine why there was no relationship between high-fat diet and anxiety. We found some complications in our study. In the Morris water maze, there was no significance whatsoever, most likely due to the number of Nile grass rats that were taken out of the study. In the elevated plus maze, Nile grass rats jumped off the maze multiple times. This could have skewed the results. Lastly, in the open field test, some Nile grass rats did not move at all, and we had to throw those scores out. A larger sample size would help to get clearer results. Also, having additional tests and more time to test the Nile grass rats would have been beneficial to the study. According to an article by Nora Waltz, people show anxiety when they're in an open space. They tend to want to gravitate towards areas where they can take shelter. We can test this using the open field test with the Nile grass rats. In our test, anxiety-like behavior was displayed by the subjects when exposed to the controlled diet and bright light, and the controlled diet and dim light due to the decrease in time spent in the center zone. One subject that dealt with the elevated plus maze found that the test subjects liked the closed arms at first, and then through each trial they got more curious and were more likely to go into the open arms. We found that anxiety correlated with diet, which affected entries into the center zone. Lighting also affected the amount of time in the open arms and entries, time and distances in the center zone. Another study was not able to find significant results due to issues with the rats as well. We also failed to give any statistically significant results due to the number of test subjects and some of the rats sinking. We'd like to thank Hope College for providing the opportunity to conduct research on live models and their provenance of proper care for the models by the means of veterinarian and staff to properly maintain living conditions. Also, we'd like to thank Dr. Gull for the novelty of the experimental study and for the supervision and conductance of the experiment.